Hey, it's Mr. Shrum, and I'm back with the mitosis meiosis tutorial for biology. And we left off at cell division. So let's talk about that. Cell division is crucial, a very important part of reproduction, growth, and development of organisms. So we're going to look at two main types, mitosis and meiosis. Mitosis and the cell cycle. Mitosis is a type of cell division that occurs in many different kinds of cells. Humans have specialized skills that perform mitosis to replace and repair organs and tissues and to build new structures as the body grows. Fingernails grow and wounds heal because of mitosis. Once a cell reaches its maximum size, as big as it can get, it splits into two daughter cells that are genetically identical to the parent cell. Each cell carries the same set of chromosomes, and chromosomes are a strand of DNA that contains genes and is located in the nucleus of a cell. The cell cycle begins as a new cell forms. The entire cell cycle can be divided into these three phases. We have interphase. The cell grows and prepares itself for mitosis. Mitosis, the nucleus of the cell divides, creating those two daughter nuclei. Cytokinesis. That means the cytoplasm divides, creating two separate daughter cells. Let's examine each of these phases in detail. So we get a diagram here. And on the next slide, we're focusing on interphase. Interphase is when a newly formed cell spends the most time, and it itself has three phases. So there are three phases, and the first phase has three phases, okay? So keep that straight. G1 phase, the cell enters the G1 phase immediately after it forms through division. It starts growing and functioning normally. During this phase, the cell makes certain proteins required for DNA replication. S phase, the next phase, we have the cell continuing to grow. The DNA replicates to double the amount of genetic material, and this process ensures that when the cell does split, each daughter nucleus receives exactly the same number of chromosomes. So the daughter cells are identical copies of the parent cell. And then you have G2. And this phase is the last phase of the first mitosis phase of the whole shebang. And here the cell continues to grow and create proteins for mitosis. It also makes copies of cell organelles so they can be distributed evenly between the daughter cells. When the cell splits into two, each cell will have identical organelles. So now let's get into the mitosis and cytokinesis. And we can click each label to learn more about the steps. So interphase G2. The cell in interphase G2 is ready to undergo mitosis. The chromatin is a relaxed form of DNA. And within the nucleus, the chromatin replicates and the organelles duplicate by the end of this interphase. Then you move on to prophase. Prophase marks the beginning of the mitotic phase. During prophase, the genetic material condenses into tightly coiled chromosomes. The chromosomes contain all of the genetic material for offspring. Each chromosome consists of two identical copies or sister chromatids attached to each other by the centromere. And the nucleolus and nuclear envelope disintegrate to prepare for cell division. Mitotic spindles are made of microtubules, which help break apart the chromosomes during division. So interphase G2 into prophase into metaphase. And here we have a really short phase. A microtubule attaches to the centromere of the chromosome. At the same time, another microtubule attaches to the same centromere from the opposite side. The chromosomes align themselves at the center of the cell, and they need to align correctly to ensure that both daughter cells are exactly identical. Then we have anaphase, and here we have the microtubules shortening and then pulling the sister chromatids to either side of the budding daughter cells. Each chromatid travels toward the opposite pole. The cell elongates and gets longer and longer, preparing for division. Then you have telophase. And this is when a nuclear envelope develops around the split chromatids at each pole, and that forms two nuclei. The chromosomes uncoil to form chromatin, and the microtubules disappear. Nucleoli reappear within the two nuclei. And then lastly, we have cytokinesis. That's the last phase in the cell cycle. The cell enters the phase immediately after telophase. During this phase, the cytoplasm divides to create two nucleated daughter cells. In animal cells, the cell membrane pinches inward and downward, not downward, and down the middle of the cell. This process produces two daughter cells, each containing a nucleus. The place where this division occurs is called the furrow. In plant cells, a cell plate forms, dividing the cytoplasm between two cells. This cell plate later forms the new cell wall. The daughter cells are smaller than the parent cell because the cytoplasm has been divided between the two daughter cells. Each daughter cell begins a new cell cycle, starting again from interphase. And it just keeps going on and on like that. Regulation of the cell cycle. So how is this regulated? Cyclins are proteins that regulate this cycle. Cyclin-dependent kinases, or CDKs, are enzymes that bind to cyclins to form complexes. And these things that we call complexes drive the cell through checkpoints to successfully complete the cell cycle. And we'll discuss each one right now. G1 checkpoint. This occurs towards the end of G1 phase. Here, a cell checks for cell size, DNA damage, and growth factors. A cell that passes this checkpoint completes the cycle most of the time. If it fails, then it enters a resting phase called G0. So it's not allowed to go past this point if it fails. This next checkpoint, G2, happens towards the end of the G2 phase. Here, a cell checks whether the DNA has doubled and is undamaged. The cell repairs the DNA if it's damaged, or dies if the damage is irreparable. And then lastly, we have the M checkpoint. This checkpoint occurs at the metaphase stage of mitosis. Here, a cell checks whether the microtubules have attached to the centromeres. If they are not attached, then the cell stops until all chromosomes are eventually attached. Stem cells, have you ever heard that in the news? Well, stem cells are unspecified cells that can later form specialized cells. So they're like blank slates, and then you can form them later. And these cells have the ability to divide mitotically through mitosis, that's what that means, to produce similar unspecified cells. Ongoing research on stem cells suggests that these cells can help treat and prevent many diseases. There are two types of stem cells. We have embryonic stem cells and adult stem cells. And here we get a diagram. Stem cells grown in labs can later specialize into various tissues. So it's very cool. A very natural solution. Embryonic stem cells come from embryos grown through fertilization of eggs by sperm under lab conditions. Unused embryos available at fertility clinics also provide embryonic stem cells. These stem cells have a high regenerative capability and are used to grow new cells in a lab. Embryonic stem cells contribute extensively to research and stem cell therapy. And then we also have adult stem cells. And these are undifferentiated cells found in adults that have a regenerative capability as well. Some sources of adult stem cells are bone marrow, blood, teeth, and the liver. But these stem cells are available only in limited quantities. They evolve into tissue that's similar to their source. Additionally, adult stem cells do not grow well in lab conditions, and it's difficult to isolate them. Okay, so we have a question. Although both adult stem cells and embryonic stem cells are used for research, embryonic stem cells are of particular interest among researchers. Which medical advancements are possible using these stem cells? What ethical issues could arise from using embryonic cells as opposed to adult stem cells? And I'll show you a sample answer. Embryonic stem cells are plentiful and they can create every cell in the human body. Researchers can use these cells to create tissues. They can possibly grow new organs in a lab to treat diseased organs. Adult stem cells are rarer in the human body and they're limited in the types of cells they can make. So embryonic stem cells are just more versatile. And embryo can't grow without its stem cells. So people have ethical concerns about their use. Adults can provide consent to donate their stem cells. So there are fewer ethical issues regarding the use of adult stem cells. And that goes back to whether you consider an embryo a living being that has legal rights. And that's when science gets a little touchy. So I'm just going to move on. Haha. But you have to think about these hard issues sometimes in science. Because science doesn't tell you what to do all the time. So we talked about mitosis. Then we have meiosis. Now let's discuss the other types of cell division. Or the other type, because there's just one other type. Meiosis. In sexual reproduction, meiosis occurs in cells that create gametes. It's similar to mitosis in many ways, but there are two key differences. First, each parent cell creates 4 cells instead of 2 at the end of meiosis. So in this diagram, it makes it really clear. 1 cell divides into 2 in mitosis, but for meiosis, you end with 4 cells. And these 4 cells are not genetically identical to the parent cell because they have half the number of chromosomes that the parent cell has. And you can see that too. So this looks exactly like the end product. These do not look like the first parent cells. Humans have 46 chromosomes. And at the end of meiosis, 4 haploid human gametes have 23 chromosomes. At the end of meiosis, 4 haploid human gametes have 23 chromosomes. Male and female haploid gametes fuse together to form a diploid zygote that has 46 chromosomes. So humans have 46. And after this process, you end up with 4 having 23 chromosomes. And then the male and female haploid gametes fuse together to form one diploid zygote that has 46 chromosomes. And if that's confusing, the next few slides should clear it up for you. So here we have the different steps, and it's a 2-step process here. It starts with a diploid parent cell, denoted as 2N, because it has a complete set of homologous chromosomes. Homologous pair consists of a chromosome from a maternal source and a similar chromosome from a paternal source. Meiosis separates these homologous pairs. And the first step, Meiosis One, creates 2 haploid cells. So we go interphase, homologous chromosomes, splits up. And those are denoted as N. So actually, we'll just use this here. This is easier. So here we have 2N, and then those split up into N. 2 haploid cells, noted as N. The cells are haploid because they no longer have homologous pairs. And you see how this one has 2 and then 2. And then once those split, those are not homologous anymore. Those are haploid, if you want to get technical. Each haploid cell only has half of the parent's genetic information. Then during the second step, meiosis 2, the chromatids in the 2 haploid cells separate, which results in the formation of 4 haploid cells. So you're starting out with homologous chromosomes, those split up, and then those split up again. So bam, step 1, bam, step 2. And that results in 4 haploid cells. So let's look at it in even more detail, like Mitosis One. So we have interphase 1, the diploid cell in interphase 1 is ready to undergo meiosis 1. The genetic material here replicates and the organelles duplicate by the end of this interphase 1. Then we have prophase 1. This marks the beginning of meiosis. During prophase 1, the genetic material condenses into tightly coiled chromosomes. The homologous chromosomes align with one another. An important step called crossing over occurs as the pairs exchange sections of DNA among the chromatids. This process creates unique combinations of genes in each chromosome. The nucleolus and nuclear envelope disintegrate. Mitotic spindles form consisting of microtubules. Then you have the metaphase. During metaphase 1, a microtubule attaches to one chromosome and another microtubule from the opposite end attaches to the second chromosome in the homologous pair. The homologous pairs align at the center. Then you have anaphase. The microtubules shorten, separating the homologous pairs from each other. The chromosomes from each pair move toward the opposite poles. They're starting to split apart. Then you have telophase 1. And here we have 2 nuclear envelopes enclosing the chromosomes at the opposite ends. And in some organisms, the chromosomes uncoil and relax. Although in others, this is skipped since cells will soon go through another round of division, meiosis 2. The spindle fibers disappear. And then lastly, you have cytokinesis. The cell enters cytokinesis immediately after that telophase. And here, the cytoplasm of the cell that contains two nuclei divides. Each daughter cell contains half the number of chromosomes, so each daughter cell is haploid. During the entire process, the sister chromatids in a chromosome remain attached to each other. Meiosis 2. Prophase 2. So this second step starts off with prophase 2. There's no DNA replication before this. The genetic material once again condenses into chromosomes during prophase 2. And in this phase, there are no homologous chromosomes to align. So the crossing over of chromosomes does not occur. Each chromosome consists of two chromatids that are non-identical because of the crossing over that occurred during that first phase. Additionally, a spindle apparatus forms during this phase or stage. Then you have metaphase 2. The microtubules attach to the centromeres of the chromosomes from the opposite sides. And then anaphase. The microtubules shorten, splitting the centromeres. This process pulls the sister chromatids away from each other and toward the opposite poles. And then you have the telophase. The nuclear envelope encloses the chromatid, the spindle apparatus disappears, and cytokinesis is when the cytoplasm divides, creating 4 haploid cells. Each haploid cell is either a sperm or an egg. Each of the 4 haploid cells has unique genetic material. That brings you to a frog cell division video. I'll upload this somewhere else in Canvas because I can't show it here right now. And then there are questions to follow. And here's the explanation. See how the chromosomes from a sperm and egg are added together in a fertilized egg. For meiosis, and then it goes on to mitosis. Sample answer for B. Frogs go through meiosis to form sperm and eggs. These cells are haploid gametes, and the fertilized cell is diploid. Then the organism must go through mitosis to build new tissues. And then for part C, frogs reproduce sexually. They require sperm from a male and an egg from a female to produce offspring. This type of reproduction allows for greater genetic variation, but organisms that sexually reproduce need to reach sexual maturity before they can reproduce. They also use energy to find a mate. Then we have part D. So here we go. Chimeras are interesting organisms that have 2 different sets of DNA. They occur when 2 different zygotes fuse to form one organism. Some chimeras have no outward symptoms, while some have signs such as 2 different eye colors or 2 different blood types. This model shows how chimera form. What role do mitosis and meiosis play in the process? And here's a sample answer. Meiosis creates haploid sperm and an egg that fuse to form 2 zygotes. When the zygotes fuse into a single embryo, the process of mitosis allows the cells to divide. This process creates an embryo that has 2 different types of genetic material. So that's that for the frog cell division video. And that's it for this lesson. And next time I'll finish up with alternation and generations. And until next time, see you. See you guys. Bye-bye.